In this week's lab, we will examine spermatogenesis in the grasshopper testis and oogenesis in the cat ovary. The testis of a mature male grasshopper consists of a series of elongated banana-shaped testicular lobes. This longitudinal section shows 4 testicular lobes. Each testicular lobe has a rounded apical end and a more elongate basal end that opens into the vas deferens. Notice that each testicular lobe is subdivided into smaller compartments called testicular cysts. All the cells in one testicular cyst are in the same stage of development. In a single longitudinal section of a grasshopper testis, you should be able to find all of the stages of meiotic development, arranged in order as you progress from the apical end of the testis toward the basal end. Examination of cysts at higher magnification reveals cells of different sizes. Testicular cysts at the apical end contain small spermatogonia. These diploid cells undergo mitosis and give rise to all the remaining germ cells in the testis. As we move toward the basal end of the testicular lobe, toward the left of this particular view, notice that the cells increase in size. These larger cells are the enlarged primary spermatocytes. These cells are the ones that are committed to undergo meiosis.
So first division is going to give you primary spermatocytes.
Cysts to the immediate left of the primary spermatocytes contain smaller secondary spermatocytes. To the left of the secondary spermatocytes are even smaller spermatids. Cysts at the basal end of the testicular lobe, located on the very far left, contain sperm undergoing the process of spermiogenesis. Recall that during spermiogenesis, a spermatid ejects most of its cytoplasm and grows a flagellum. Let's take a few moments to review the concept of ploidy by identifying whether each of the following cells is haploid or diploid. Spermatogonia. If you said diploid, you would be correct. Primary spermatocytes. These two are diploid. Remember, spermatogonia enlarge and differentiate to form primary spermatocytes. Meiosis has not yet begun. It is the primary spermatocytes that are destined to divide by meiosis. Secondary spermatocytes. These cells are haploid. Remember, meiosis 1 is referred to as the reduction division. Spermatids, haploid. Spermatozoa or sperm, these are haploid as well. Let's turn our attention to oogenesis, egg production in the female. The ovary of a mature cat is subdivided into 2 regions. The central region, or medulla of the ovary, contains numerous blood vessels, while the outer region, or cortex, houses a large number of sac-like follicles in various stages of development. Let's enlarge the cortex region and examine the various types of follicles it contains. Notice that there is a high concentration of tiny follicles located just under the surface of the ovary. These peripherally located follicles are known as primordial follicles. Each primordial follicle contains a primary oocyte, surrounded by a single layer of squamous cells. Think back to your animal tissues lab. What does the word squamous mean? If you said flattened, you would be correct. Notice how flat the cells are that surround each of these primary oocytes. Where, might you ask, are the oogonium, the diploid cells that enlarge to form the primary oocytes? In cats, like other mammals, including humans, all oogonia formed during embryonic development of a female have been converted to primary oocytes by the time she is born. This is one of the reasons why females who have gone through menopause cannot have children. They have depleted their egg supply. Males, on the other hand, can continue to father children until a ripe old age, because spermatogonia, unlike oogonia, are able to replace themselves via mitosis until the male dies. Slightly larger follicles, like this one, are known as primary follicles. They differ from primordial follicles in that the primary oocyte is surrounded by a single layer of cube-shaped cells. If the primary oocyte is surrounded by two or more compact layers of cells, then the follicle is called a secondary follicle. This secondary follicle possesses 3 to 4 layers of cells. Notice the dark pink ring indicated by the black arrow. This is the zona pellucida, a glycoprotein membrane that surrounds the plasma membrane of the oocytes of mammals. It is the zona pellucida that contains the species-specific sperm receptors. As secondary follicles continue to grow, fluid-filled gaps begin to appear between the cells. At this point, the secondary follicle becomes known as a tertiary or antral follicle. A fully mature tertiary follicle is called a Graafian follicle. Note the size of the fluid-filled antrum. A few hours prior to ovulation, the primary oocyte completes meiosis I, forming a large secondary oocyte, seen here still enclosed in the zona pellucida, and a smaller first polar body, which cannot be seen in this view. The ring of cells surrounding the secondary oocyte is called the corona radiata. At ovulation, the secondary oocyte, along with its zona pellucida and corona radiata, will be expelled from the Graafian follicle. It is worth noting that the secondary oocyte will not complete meiosis II, forming an ovum and second polar body, unless it is penetrated by a sperm. In humans, sperm penetration, more properly known as fertilization, typically occurs in the oviduct of the female. What happens to the Graafian follicle once ovulation has occurred? It is transformed into a structure known as the corpus luteum. Notice that this cat ovary contains 2 corpora lutea.